Thank you for the introduction. Again, thank you for the opportunity to be presenting here today. In this study that I'll present today, just recently got accepted on JDS, so it's available online for some of the more details that I won't have time to go through today. If you've seen my talk before lunch, it will be very similar to introduction because the concepts are the same for where waste milk is coming from. But again, anytime we treat cows with antibiotics, one concern that we always have is the potential for these antibiotics resulting in drug residues in the milk to the point where it will not allow this milk to be sold for human consumption. And this is commonly called waste milk. Again, this could be coming from not just because of a microbial treatment or high somatic cell count or any other factor that the farm deems the milk to be of lower quality and should not be commercialized. Similarly, as I talked earlier about in the US at least, 1 in every 3 farms, we have information that shows that they would feed waste milk to pre-weaned dairy calves. Again, when you look at reports for different countries and just with talking with different people, I hear reports much higher. And even in California alone, I also see that being reported at higher rates. One of the reasons why farmers do this is we have evidence for economical savings. So this is a study that I'm showing here from Sandra Godden, where she compared the cost of feeding waste milk, pasteurized waste milk versus milk replacer. And from that, she saw about saving of 70 cents per calf per day while feeding waste milk instead of milk replacer. Again, one of the concerns that we have is potential for selection of antimicrobial resistance. The EFSA has recently released a report that actually is a nice overall review of some of the studies that have addressed this concern and also talked about alternatives for waste milk if we're not feeding it to pre-weaned calves. I'll also give a little bit of some background information on some studies that I've conducted on waste milk. Again, this is actually one trial that I talked earlier in my previous talk, but this was one study we conducted where we had calves from birth all the way to weaning. It was a very controlled environment where we got whole milk, commercial whole milk from a farm, and the only difference between the treatment and the control group was that we added 4 different antimicrobial drugs at concentrations that we have previously found in waste milk from dairy farms in upstate New York. Shortly, what we observed is that after usually week 2 and all the way through the end of the study, we found that calves receiving these drug residues, and again, we were collecting weekly samples and culturing for E. coli. We observed that these E. coli had significantly higher proportion of resistance to beta-lactams and tetracyclines, to cephalosporin drugs, and also a higher proportion of multi-drug resistant E. coli. Another study that we conducted for those same samples at a weekly basis is that we actually looked at how does this affect the microbial composition of that bacteria. So we ran a 16S RNA. From that, we did observe that feeding waste milk, which is the red box, resulted from this discriminant analysis, resulted in a microbiota that it basically matures quicker to a more older animal compared to calves that don't receive these drug residues, where we see a more gradual progression of that microbiota into a final, more mature microbiota that we observe in older calves. And earlier today, too, I talked a little bit about how this feeding waste milk to calves could potentially affect the function of the microbiota. So the objective of this study that I'll present data today was actually to just do an overall screening to identify antimicrobial drugs as well as their concentration in dairy farms in California. We also conducted a short survey at a herd level to try to understand factors that could be related to why we are finding these drug residues on this waste milk. We also, from the milk samples, we did some bacterial aerobic culture and anaerobic culture. And also for E. coli, whenever we identified E. coli in the milk, we did a further testing for minimum inhibitory concentration for MIC testing using Kirby-Bauer disk diffusion. Just for a background, so California is the yellow state there in the US. So all the way in the corner of the West, we have about 1.7 million dairy cows. We're the biggest dairy producer and also the biggest state in number of cows in the US. So this is just briefly what I was talking about. We had about 25 total dairy farms where we collected one sample, so it was a cross-sectional study. We measured the microbial concentration and presence in the drug using liquid chromatography, mass spectrometry. We had the on-farm questionnaire, and we also conducted some microbial testing on these milk samples. This is just some descriptive data information for that. If you aren't from the US, especially in California, you won't know the different counties, but basically Tulare, Glen King, those are very large dairy counties in California. They call it the Central Valley. These samples were convenient sampling, so just according to relations we had with the farms or with people who were helping us collect these samples. Related to farm size, most of our samples are collected from dairies with 1,000 lactating cows or more, and that's usually what we typically find in California, more towards larger dairy farms. Some more information that we collected was how many cows on average are you milking into the bull tank? So from that, we had farms said that we milk usually 1 to 9 cows or 10 to 29, 35, 59. Overall, we found that most farms reported about 10 to 29 cows. And that kind of helps us just have an idea of how much waste milk are they dealing and how much waste milk they have to get rid of at the end of the day. Another question we have was, is waste milk being fed to calves on your farm? So 5 farms, 20% raised calf heifers off the farm. And because of that, we couldn't collect that information. So they do have it commonly in California. They'll just send the calf somewhere else. They'll raise the calves for them, and they'll get them back later on, and they'll pay for that cost. For the remaining farms, all farms except one did not raise their calves or use waste milk as a feed. And this one that didn't use waste milk, it actually sold the waste milk to someone down the road, and they fed it to the calves. So that was the final destination anyways. And for the farms that were feeding waste milk, about 75% were pasteurizing this waste milk. 25% were just feeding it straight on raw. Okay, so this table shows you some initial results for what we observed for drug residues in the milk. So from the 25 samples we submitted, for at least 60%, we found at least the presence of one drug residue. The screening that we used was screening for 27 different drugs, 26 being antimicrobials, and 1 was for fluniximaglamine, so an anti-inflammatory, non-steroidal anti-inflammatory. When we looked at the distribution by drug class, the most common drug residue by class observed was for beta-lactams, followed by tetracyclines and sulfas. We also found some samples that had both beta-lactam and tetracycline drugs in them, so that would be one sample, about 4%. When we broke this information further down within individual drugs, we observed that the most common drug residue we observed was for ceftriaxone, so a third-generation cephalosporin. Secondly, we observed most commonly these was for residues related to cefepime and oxytetracycline. And here I'm also presented the mean concentration that we observed for these drug residues in the milk. The standard error and the tolerance is what the FDA, the Food and Drug Administration in the US, has for the cutoff that anything above this amount of drugs cannot be sold for human consumption. So that's just as an overall reference. For the culture results that we observed, so from the 25 samples that we submitted, about 21 or 84% we observed the growth of Streptococcus. And following that, the 2 other most common bacteria we observed was for Staphylococcus species and E. coli. I'd like to highlight on this one that we did find for 2 samples or 8% mycoplasma. And I always highlight that to farmers about it's important to pasteurize and process samples, but if you're feeding this milk raw, feeding this milk with mycoplasma, you'll see a lot of problems in the herd that go way beyond just concerns with antimicrobial resistance. And again, for E. coli, we actually did further testing to look at susceptibility to different drugs. For the 10 E. coli we collected, or about 40% of the samples, we observed that from those, 30% were resistant to ampicillin, 20% cephalothin, and 10%, actually 1 out of 10 for ceftriaxone. We did observe some resistance to tetracycline and to sulfoxazole. And this just highlights, because this is almost a separate problem about not just the drug residues that we could be feeding to calves that could be affecting the microbiota and selecting for resistance. These would be just bacteria that are ready to go, they're already resistant, they're present in that waste milk. We don't know if the residues in the milk were helping select for them or they're coming from some other source, but these bacteria could be directly fed to calves as they are. Another question that we had, just to try to better understand where are these drug residues coming from for these samples. So this was just a herd level question related to what is the most common drug to use to drying off a cow, or for mastitis, or for reproductive disease, or for lameness, or for bovine respiratory disease. Not surprisingly, ceftriaxone was the most common. In the US, for the ceftriaxone drugs we have, most of them we have a 0 milk withdrawal period. So it is their drug of preference a lot to reduce any concerns with the milker milking those cows with potential drug residues into the bull tank. As I said, for most of these, there's a 0 withdrawal period. However, for the mastitis tube that we have, that would be an intramammary treatment, we do have a withdrawal period of 72 hours. And for this study, that's where we believe that most of those ceftriaxone residues are coming from in waste milk, would potentially be from cows treated for mastitis using ceftriaxone. Okay, so overall conclusion was that we observed that ceftriaxone was the most prevalent detectable drug residue, followed by cefaparin and tetracycline. I actually did a survey study like this in upstate New York. Not surprisingly, ceftriaxone was also the most common drug residue there, so it kind of matches that tendency I have observed before. Multiple bacteria species were cultured from waste milk. Again, that highlights the importance of pasteurizing this milk prior to feeding them, especially for bacteria that we may have a lot of challenges treating in calves, like mycoplasma later on. And again, the most common drug used to treat cows overall on the farms that I sampled was ceftriaxone, followed by penicillin gene tetracycline. And as I highlighted, ceftriaxone is probably, for intramammary treatment, that's where we probably expect to see those residues that we detected in the waste milk. Okay, that's all that I had to present for today.